Thank you very much, Ricardo. Thank you all so much for being here. As Ricardo mentioned, if you couldn't hear or see it up here, Sam Scarpino, I'm the Director of AI and Life Sciences at Northeastern University. I was previously here in the Network Science Institute before leaving for about 2 years at the Rockefeller Foundation. What I'm going to talk about today is the issue of ground truth in biology, and most of the talk I'm going to try to convince you that the problem is widespread and represents a number of significant challenges for applications of machine learning and artificial intelligence to living systems. And then I'm going to try to convince you that there's a path forward. So a bit of motivation. These are data from individuals with what was up until recently an incurable form of cancer called multiple myeloma. And the data come from 20 individuals who are part of a cohort study where they're sequencing the RNA to look at gene expression for individuals at different stages of cancer. So each row here is a different RNA sequence being expressed by a gene. There's about 60,000 rows, and so we've got like 20,000 genes, but they have different ways of starting and stopping inside of each gene, splicing themselves together, et cetera. So from those 20,000 genes, we have more than 20,000 mRNA products. And the dark spots here are places where the genes' expressions are correlated with each other. So if you're used to looking at these kinds of things, what should jump out at you here is that there's a lot of structure in the interaction network here. You see lots of little black dots off of the diagonal line. So the rows and columns are the same gene. So this is diagonal here is the same genes correlated with each other. And there's lots of hierarchical patterns, which is the way our bodies are supposed to work, right? So all of our genes interact in a hierarchical regulatory network. And so we would expect to see these sorts of tree-like structures that give rise to this blocky pattern here. What we know is that 20 to 30% of the individuals in this sample will not respond to any form of treatment whatsoever. The rest will, at least for a period of time. But there are no genetic markers associated with individuals who will or will not respond to treatment. So there's no way to tell by sequencing the genome of the tumor whether the individual will be in this group that will respond to the treatment or not. However, if we look at these same 20 individuals after all of them have stopped responding to treatment, and we analyze the same RNA sequencing data, so looking at gene expression, the rows and columns are in the same order as they were on the previous graph. It's the same individuals. They've just now stopped responding to treatment. What you can notice is that this figure looks very different from the other one. May not be able to explain exactly why. I can tell you what has happened here, which is going from responsive to non-responsive, we see a complete breakdown in the group structure, the pathway structure, the hierarchical organization of the regulatory systems that are present into something that looks much more like a hub and spoke model of regulation, where you have one or a small number of genes that are regulating a large number of genes. And we know that this is a hallmark of cancer. This is how it's avoiding treatment. This is how it's replicating so quickly and avoiding aspects of the immune system. So while there are no genetic markers, it doesn't mean that there aren't signatures associated with this key phenotypic change, meaning a lack of responsiveness to treatment. And it also doesn't mean that we don't have mechanistic explanations for what's going on. So this is work that was done by Dr. Brennan Klein, who did his PhD at the Network Science Institute and is now a postdoctoral researcher there. And what Brennan was interested in is how, if you have regulatory networks, so imagine these are genes interacting with each other, from two different microorganisms, and this microorganism here ingests this one. Maybe this is like a mitochondria or a chloroplast, and we now have an organism that was a prokaryote, becomes sort of a proto-eukaryote, and then a eukaryote, meaning that it's got other little organisms that replicate along with it in a very stable life cycle. We can look at the pattern of gene expression and interaction as it shifts from this prokaryote-like cell to this eukaryote-like cell. And what we can see is that the pattern mimics what happens with cancer. So effectively what's going on inside of a cancer as it progresses is the loss of the regulatory patterns that are important for eukaryotic life. So again, no ground truth in the sense that there isn't a consistent signature that you can use to identify individuals who do or do not respond to treatment. But there are other pieces of information that we might be able to bring to bear on the problem. And, of course, you might imagine then, well, if this is the hallmark of cancer progression, are there then drugs that we could target to effectively prevent the network from degrading along this gradient here? So my claim, and a big part of the reason why I'm here, is that I am convinced the biggest open question right now in science and society is how predictable are living systems? So if you're a scientist, how predictable something is will relate to your ability to study it. If you're a practitioner, how predictable something is will relate to your ability to interact with it, control it, etc. And we don't really know how predictable living systems are. We have some qualitative ideas about it, and we have some individual examples. And where I think most people come down, and this is what I want to try to convince you is not the problem, most people tend to come down that the problem to addressing this question, in fact, the problem to improving predictability in living systems, is that we just don't have the right kinds of data. If we had better data, we would be able to predict what's happening in these living systems, and it's really just a data problem. If it is a data problem, we actually have bigger issues at hand than that. And it turns out that this question, I think, is at the heart of what we deal with in machine learning and artificial intelligence, and at the heart of what we mean by ground truth. And this should be a fairly simple question to answer, but turns out it's actually rather difficult to get people to agree on what a definition of data. In fact, we can't even agree on whether it's singular or plural. I'll probably switch back and forth. But is anybody feeling brave and want to give a definition of data?
Set of values which are not currently forming any patterns or all, like unstructured, structured, and different types of data are there.
So it's a set of values, and it might be structured or unstructured or have all sorts of other attributes associated with it. Okay, what else?
Generally a quantitative thing.
Okay. Often it's either quantitative or something that we want to do computation on. No other definitions?
Maybe the key drivers of what we want to predict or...
Okay. What we want to predict. If we just look this up in the dictionary, it says it's factual information such as measurements or statistics. Now, if anybody in the room or online has a background in philosophy, if we start something by saying it's factual, that's going to create a whole lot of problems for us down the road, right? Because that means as soon as I say something is data, I've stopped worrying about whether it's true or not. I've decided that it is real. Information in digital form that we could transmit it or processed, all of these things are definitions of sort of the things we do with data as how they are defined. But quite often, if you look up definitions of data and if you really try to pin somebody down, they're going to call it a fact. And in point, if you look at how Bayesian statistics operates, you have a function that takes data as input, puts it through a likelihood equation, and returns something out the other side. But as soon as you put something into that part of the equation that says data, you've admitted that this is true. You can have noise associated with it, you can have observational bias, et cetera, but you put it into this equation, and then that becomes the reality. And this, of course, has led, this is from 2000, but this, of course, has led many people to just say, well, maybe we don't really even need models for anything anymore. We just ask the data, right? So this is a sort of end of theory data deluge makes a scientific method obsolete. And this was almost 30 years ago now. And of course, that has not been what has come to pass, especially in the life sciences. So what are we going to try to do today? I'm going to say just a little bit more about machine learning and data. I'm going to talk about the problem of ground truth as it pertains to supervised learning. I'll tell you what I mean by that in just a second. Then unsupervised learning, and then I'll talk at the end just a little bit about experiential AI. So I feel like it's probably a requirement now to include something from ChatGPT in the talk. And so I just asked it what machine learning is, and it says machine learning is a type of artificial intelligence. In fact, you probably couldn't even get everybody to agree that that's true. A lot of ML people would say it's not AI. A lot of AI people would say it's definitely not ML. That provides computers with the ability to learn without being programmed. It focuses on the development of computer programs that can access data and learn for themselves. Computers are able to learn from data, identify patterns, et cetera. So some of the less controversial aspects of this, I think, are actually fundamentally the most challenging here, which is it's all about learning from data and then being able to generate something that we care about off of those data. But nowhere in this definition, and it got this definition off of Wikipedia and all the intellectual stuff that's been written about data, nowhere does it really wrestle with how do we know that the data we're collecting and operating on are actually meaningful and representative of the thing that we care about. Like, how do we evaluate data? So just very quickly, this is a nice review article on current challenges for deep learning in the biosciences. And they look at two different kinds. They talk a lot more about other kinds, but two main kinds of machine learning tasks that we're engaged in. And I'm going to bucket the talk into these two broad. One is supervised learning, and this is where we have a labeled data set, right? So I have these cancer patients. I know which ones are responding to treatment. I want to try to predict which ones are responding to treatment or not using gene expression data. So I have a labeled data set. The second is I have an unlabeled data set, and I call this unsupervised learning. So maybe instead I just give you a giant text file of gene expression data, and I want you to tell me whether there's anything interesting going on here. And maybe what comes out the other side is that there are two clusters of individuals, and later on we can figure those clusters relate to responding and non-responding patients. But you could basically think about, I have some labels on my data, I don't have labels on my data. Okay, so challenge with supervised learning. The first example, can we use data to predict epidemics? This is a question that Giovanni Petri, who's a principal research scientist at the new AI institute in Italy called Sentai, has been interested in. We've been interested in this for years. And we're interested in part because of the question, can we predict this time series? This is a time series of influenza cases in Texas from 1930 to 1960. If you're not familiar with the state of Texas, it's geographically about the size of France, but there's only 25 million, probably 30 million people that live there now. So it's densely populated in some areas and incredibly rural in other areas. And the problem with answering this question of whether we can predict this time series or not is that we're not going to get past the stage of arguing with each other over how we're going to predict it and how we're going to evaluate the prediction. Right? So one of the ways we might predict this is to fit a mechanistic model, like an SIR model if you've heard of these, to the data. However, I have my favorite model for flu. I'm sure most of you that have worked on flu have your favorite model, and we're immediately going to reach an intellectual impasse where we will refuse to agree on each other's models, and more importantly, we're not going to agree on how to evaluate the forecast. Maybe I care about the root mean squared error and you care about the log likelihood. And maybe I'm going to do an 80-20 split and you're going to do a 70-30 split. Or maybe I'm going to hold out 10% and never do any training and testing until the very end. But my 10% is going to be different than your 10% because I only care about the peaks. I think that's harder to predict. And maybe you care about the noisier parts down here. And sure enough, my model is going to work the best on the things I care about, and your model is going to work the best on the things you care about, and we're not ever going to get an answer to this question. So instead, Giovanni and I took an approach to say, what if we just abandon the models, abandon the metrics, and we use information theory, and just say, how much information is contained in this time series? Is there a model that exists somewhere in the universe of models that I could use to predict what's happening in this time series? Maybe I can't ever figure out what that model is, but is it out there? And if it was out there, how good in theory could I do? The specific tool is called permutation entropy. Because it's a discrete form of entropy, the notation is a disaster, but it's actually not that complicated to reason through it. So imagine my time series I want to predict has just gotten a little bit easier. It's zeros and ones, or it's coin flips. And I'll do this in 2 dimensions. So you have to pick how many dimensions or how long of a window you want to use. And so I'll just count up down the time series. Okay, I have 1, 0, 1, I have another 0, 1, I have a 1, 0, I have a 1, 1, and I have a 0, 0. And I make a a little histogram or bar plot over the number of times I observe these different symbols. And the flatness of this, or the Shannon entropy, is the permutation entropy. And 1 minus that is how predictable it is. So the flatter this distribution is, the less predictable it is. And so the way to think about the predictability here, if I have a fair coin that I'm flipping, I can, of course, learn a model, a mechanistic model that will generate that time series. But I won't actually be able to forecast the future any better than just sampling from the historic data I used to fit the model. Whereas there are many other kinds of models, like maybe a sine wave, for example, where as soon as I learn the parameters of that sine wave, I will be able to forecast out into infinity far better than sampling from the historic data. You could think about this in slightly higher dimensions. So imagine I have something that looks like an epidemic curve. And instead what I'll do is I'll say, OK, this symbol here will be flat, flat. This will be flat, up. This will be up, up. This will be up, down. This will be down, down. And you can have it be big up, little up, et cetera, et cetera. So what you do is you define your window size. You decide if you care about magnitude or not. And then you can generate all possible combinations of symbols that could occur under that size. And importantly, and I'm going to do this unfortunately a little bit throughout the talk, which is I'm not really going to explain things clear enough, which is not a very good example of how to give talks, but we're going to do it anyways. So importantly, Garland et al. In PRE showed that this sort of somewhat arbitrary kind of why on earth should this work symbolic counting of things in time series, what they showed is that this actually works for models where we know how to calculate the predictability. So there are plenty of methods, say for deterministic nonlinear systems, where we can actually calculate a quantity that will tell us something about how forecastable the system is, right? So if you're familiar with like Lyapunov exponents, the predictability will be a good predictor of a Lyapunov exponent. Importantly, the predictability here doesn't require that it's deterministic, and it doesn't make certain assumptions about the stability of the model structure. And so there are a lot more places where you're allowed to apply this than you are for Lyapunov stability. You also can do this pretty easily on your laptop with some code, as opposed to what you would need to do to actually get an estimate of some of those other quantities. OK, so let's return to the question at hand here. Can we predict this time series? And so the way that I'm going to do this is I'm going to say I'm a Bayesian. We're all Bayesians, whether you believe it or not. And the way that we learn is we see more data and we learn things. So what I'm going to do is I'm just going to calculate the predictability, say, up through here, counting up the number of different symbols that show up. And then I'll do it up to here, and I'll just keep sliding forward. And so what we'll end up with is a predictability measure as a function of how much data I'm feeding in to that engine. So predictability, higher up here means it's less predictable. Lower means that it's more predictable. Years of data is only going to apply to the influenza curve, but I have some white noise and a sine wave, so we can kind of do a little bit of a comparison about how predictable we think it is. So one of the problems with predictability is I can't tell you what value of predictability works for your application case. And so what we'll do is we'll try to bound it by something that we know is not really predictable, which would be like just drawing random numbers from a normal distribution, and something that everybody sort of thinks is predictable, which is a sine wave. So just think in your head, like, what is this going to look like? Where's the white noise going to be? Where's the sine wave going to be? And then what's the change over amount of data going to look like? So as I calculate the predictability for white noise, just drawing from a Gaussian distribution or flipping a fair coin, how do you all think that the entropy will change as I see more data? So I'm going to give you a time series of length 10,000. You're going to calculate the entropy, and then I'll give you another one of 100,000. Is the entropy for the 100,000 going to be different, the same?
Definitely different.
Different. Why different?
Randomness will increase, then the data spread, if the data spread increases, the entropy increases.
OK.
Variation will be more in the points between that. So it will be much of change, so it will be more entropy.
OK. Any other guesses? One of the things that shows up in my course evaluations is I do this thing called read my mind, where I just keep telling you that's not the answer I'm looking for, even though it might be the correct answer. It's just not the one I'm looking for. So here's what's going to happen. It will increase slightly, but very quickly we're going to reach a state where we've seen all the possible symbols that will show up. So this is not the entropy with respect to the distribution of data. This is the entropy with respect to the symbols that we're observing. And the symbols here would be like, was there an increase in the two data points? So I draw a 0, now I draw a -1, now I draw a plus 0.05, now I draw a minus 0.05. And so we're looking at how it changes in time. And that's not going to change very much. There is a true entropy of this time series. There is an actual value of that as long as we're drawing from the same Gaussian and not changing the parameters. And we will realize that very quickly, and it will be close to 1. And your intuition is right that it will actually take infinitely long for it to reach 1. And so it will increase subtly over the course of this. For a sine wave, what's going to happen is the entropy is actually going to drop, because until we get through a full cycle of the sine wave, we're going to be seeing lots of things that look unusual with respect to the symbol distribution. And then as soon as we get through one full cycle, we won't see anything. And in fact, this will eventually go all the way to 0. I've corrupted this with a little bit of white noise because otherwise it would be way too far down. Here's what happens for flu. The entropy is initially quite low. It shoots up, and then it appears to stabilize at some value that's effectively white noise. I have yet to find, we did find one, but I have yet to find a model that you write down on a piece of paper that generates this kind of behavior. But it's all over the place in living systems. Now, you might say, maybe I cherry-picked this example. Texas is a weird state if you've ever been there. Maybe it's not really a systematic problem. Now we're looking at here our data from all 50 states in the US. I sample different starting points in the time series, and I calculate the permutation entropy for influenza. And as the time series gets longer and longer and longer, the entropy continues to climb up, 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 up. The more data I see, the less information I seem to have in terms of prediction. This does not happen in a chaotic system. That does not happen. Chaotic system has an entropy that it will go to. It might take a while, and it does not increase like this. The only other model that you can get to behave this way is one where, imagine that I have this giant model, like a large language model with 10 to the 9 or something parameters. And what I do is every time I take a draw from that model, I regularize it. So let's say I'm going to set 50% of the parameters to 0 and generate an outcome. But every time I regularize it, I do it differently. So the easy way of saying it is that the model changes, the data generating model changes in time. Physicists don't like that because it's not necessarily true. The data generating model can be fixed, and it can regularize itself differently in time, meaning different non-zero parameters matter for generating the data. However, the intuition here is that by the time I have enough data to learn the model parameters, the model has shifted, and those parameters are no longer valid. And so I actually can't learn the model fast enough to generate predictions any better than I could just from sampling from the historical data. I actually think this is a fundamental feature of living systems because they're evolving. They are actually interacting this way if you think about hosts and pathogens, et cetera. And this actually turns out to be true for chlamydia, dengue, gonorrhea, hep A, flu, measles, mumps, polio, whooping cough. If you stare at this, this is something that you're not supposed to do, which is I flip the order of the y-axis here, so this is now more predictable. Less predictable, this is more predictable. You might kind of say like, hey, if you're a physicist, you might be tempted to ask if these will collapse down on each other, and they will. And it's the first and second moment of the distribution of secondary infections that will collapse these down on top of each other. So there actually are biological explanations that account for why, say, for chlamydia, the predictability falls off so much faster. So this is a sexually transmitted infection. It will have a power law-like network of secondary infections, which in the limit means that there will be no second moment. And so what's going to happen is it's very bursty, and then you're going to have lots of sort of small chains and then bursty again, and that means that the predictability is going to degrade incredibly quickly. Whereas, say, something like measles and whooping cough, these have a very small second moment and a high R naught or first moment, and they're going to march along deterministically until they infect the whole population. Okay, So, plenty of labeled data on epidemics. Major challenge predicting them because the model structure shifts in time. What about other places? We did a project for the CDC where we were asked to take as input influenza genomes and predict something about them. Will an antibody bind? Will a drug work against it? And they had this very nice labeled data set where they put thousands of flu genomes into the lab. They test them for antiviral resistance. They test them for antibody binding. And so we're going to train a neural network to take as input a flu genome and return as output a prediction about the phenotype. So I'm going to skip over this. There's some very interesting biology about how influenza neuraminidase inhibitor resistance evolves, and so you should ask me about it later. Here are the data. So each of these little tips here is a flu virus. This is a phylogenetic tree that relates them. So like these two viruses are closely related and much further related than these viruses are here. In blue are the viruses that will respond to the treatment, and red are the ones that are resistant to the treatment. And all I'm going to do is try to train a classifier. And it turns out that it's pretty easy to do this. So here's just the sensitive versus resistant, and so all of these should be zeros, all of these should be ones, and for the most case there are. There's a few that it gets wrong, and it's kind of interesting why it gets them wrong. And again, here's this ROC curve. So we have all these stats, positive, predictive, but you can throw everything you want at it, and it's doing a really, really good job. However, what about this process called evolution that we know will drive change in the virus? What if I specifically build the training and testing exercise to evaluate what will happen in the real world, which is not random subsets of the data being thrown at me, but instead I'm going to see a particular kind of flu for a while, and then I will see a new kind of flu. So what we're going to do is we're going to train the data on H1N1 pre-2009 pandemic and test it against the post-2009 pandemic. Now the interesting thing, we know the actual causative mutation for the resistance. And it's the same in both cases. Single amino acid swap, we know exactly what it is. So you could do this with logistic regression and you would never fail. So here on the right, sensitive resistant model trained on 2009 H1N1 tested against H1N1 2009 data. And what you can see is that we're doing a better job at classifying sensitive versus resistant. Now when we test it against the post-2009 data, we're doing no different than random. The data set's not balanced, which is why they're kind of seeing it down the bottom. Same exact mutation that's causing this. Turns out what's happening is that neural networks are really good at what? What are they good at?
Understanding the complex patterns.
Yeah, but like, said even like, what are they doing?
Learning from the error basically.
Yeah, but they're doing a really good job of clustering, right? And so what it turns out it's doing is it's just memorizing where in the evolutionary tree the resistance mutations are showing up. The problem is the way resistance evolves for flu. So each of these, say, are sensitive, the red are resistant. What happens is you have sensitive lineage that shows up, resistance evolves, that lineage goes extinct, a new sensitive lineage shows up, resistance evolves. And so the clustering pattern with respect to where the resistance shows up on the tree changes in time. And instead, what the neural net has done is to memorize the tree structure, which makes it basically perfect if you're slicing the data up over time. But if you do it over evolutionary scale in terms of the training and testing, it's going to completely fail. Now, again, you might say, well, that's pretty obvious. If you just ask an evolutionary biologist, I know Professor D in the background is not making this mistake. But that's because you ask evolutionary biologists what's going on here, right? This is why we have interdisciplinary research. This is why we do the stuff that we do at Northeastern. Okay. How am I doing on time? Doing okay. All right. Although I'm going in the wrong direction. Let's fix that. Okay, what about experimentally? What about experimentally? So I want to cure cystic fibrosis by swapping in a healthy allele for individuals with cystic fibrosis. Or maybe I want to go in and I want to try to address this issue with the gene expression data for cancer patients. So what we did is worked on this data set where we looked at different combinations of antibiotic resistance mutations in different bacterial species and different bacterial genomes. So I'll explain how this works. So we have 3 different kinds of genomes. Wild type, which this just means this is the baseline, and then doesn't matter what these mean, but what this means is that they affect the way proteins will fold in these organisms. So 2 different protein folding environments across 3 different species of bacteria. And we'll look at 1, 2, 3, 4, 5, 6, 7, 8 different possible alleles with respect to antibiotic resistance. And then we'll assess how well these different bacteria grow under the presence of antibiotics. So what we're looking for basically is if I measure the effect of antibiotic resistance alleles in E. coli in the standard background of the genome, how good would my prediction be if I then tested it in a different protein folding environment, or if I put it into a different bacterial species? So, and they actually did this engineering experiment. So what we're looking at here, the big blue arrow means if I go from this allele here to this allele here, I get a big positive effect on growth. So that's good for the bacteria, that's bad for us. The dashed line here means there's no significant effect. You'll see some red lines show up, that means there's a negative effect. So the question we want is like, how good of a job could we do at predicting? It turns out that it's not just that we're going to be off by a little bit. It's not like if we take this one looks like it's the best mutation here, so I'm going to engineer this mutation into these different environments and into these different bacterial species. It's not like we're off by 10%. It grows a little bit slower, it grows a little bit faster. Sometimes the sign completely changes, so what was a positive effect becomes a negative effect. The magnitude shifts wildly. We go from significant to non-significant. So I'll just show you the full experiment here. So look at this one here. In E. coli, this is the best mutation in this genome background. It stays pretty good in Clostridium, and then when we put it into Listeria, it actually turns out to have no significant effect. We can look at this one here, which has the largest effect here, positive in Clostridium. If you don't see an arrow here, that means it died, and we actually couldn't measure the growth. This is bad. This is a big problem, right? This is what we want to do with CRISPR, is take data from E. coli in this wild-type genome and swap in different alleles into these different backgrounds. And the take-home message from this study is in order to predict the sign of the effect, sometimes even whether it was lethal or not, you have to have the exact measurement in that organism, in that genome background. Okay. So living systems, the data generating model changes, with apologies to the physicists for being a little lazy with the words there, in time, and it might change for predictable reasons, but it changes such that the ground truth is not really something that we could rely on for sort of true out-of-sample prediction. But what about unsupervised data? What if I don't care about the labels? I'm going to throw the labels away. There's no labels. Interestingly enough, there's some research in the Network Science Institute being done on labeled versus unlabeled data, and it turns out it's actually not that easy to just decide your data are unlabeled. There's actually something about your data that will tell you whether it's labeled or not, so you can't just say I'm throwing away the labels. They might still be there. Did some work with this startup called ThinkMD, where we're trying to predict pediatric malaria in a sort of unlabeled way. The way that we did this, it's a little bit of a cheat to say it's unlabeled, but basically a pediatrician wrote down a decision tree on a piece of paper, we code it up in JavaScript and put it in an app, and then somebody will diagnose pediatric malaria. So there has been some labeled training data that happened through the course of this pediatrician getting an MD and getting board certified, et cetera, but we didn't train a machine learning algorithm against a labeled data set to try to predict malaria. And the way we designed the study to test this, the actual experiment that we wanted to run was we then did a supervised learning exercise where I took a whole bunch of patient encounters and trained a random forest model to try to predict malaria. And so what we were doing is actually testing whether the physician diagnosis was better than the ML diagnosis, and more importantly, whether we could get to the same diagnosis with fewer questions, because people don't like clicking through the apps with the ML diagnosis. So we set up what we thought was this really creative way of testing it, which is Everybody gets a malaria rapid diagnostic test. Everybody gets evaluated by both systems. However, at the time, there was also a grant to see whether the app itself could read the rapid diagnostic test. And we've all used these now for COVID. And so you just take a picture and you try to see if it's positive or negative. So it turns out that we can predict it quite accurately, pediatric malaria, but there was a bit of a catch. The catch came down to how did we decide to evaluate whether the OCR we were doing on this detection of like, is this positive or negative? Is this positive or negative? How did we decide to evaluate that? Well, we decided to also PCR test everybody for malaria, right? That seems like the logical thing to do. PCR test is usually considered the gold standard. So now for everybody, it's about 6,000 individuals across 5 clinics. We're going to have a clinician diagnosis, an ML diagnosis, a rapid diagnostic test, and a PCR test. Here's what happened. They all wildly disagreed with each other. Importantly, the rapid diagnostic test and the PCR test all disagreed with each other. So here, this RDT test says malaria. So this would be like you do the thing and you put it on the rapid test and you have COVID. Then you go get a COVID test. So 50% of the time that the malaria rapid test was positive, the PCR test was positive. That's actually already not very good, right? So only 50% of the time do you get a positive on both tests. However, 40% of the time the rapid diagnostic test came back negative. If you do a PCR test for malaria, it's positive. We also have some cases, this one's actually pretty interesting, where PCR is negative and RDT is positive. So what's going on here? Well, we actually know what's going on here, which is we can look into the data, the clinical workups, and we can see that these people don't have any symptoms. And importantly, we can see that they're older, and we can assess their HIV status. And these are individuals who have or minimally suppressed HIV and have had persistent parasitemia associated with malaria infection that's not detectable via rapid test. They don't have symptoms, but they are PCR positive. There are parasites there. What's going on here is super cool, and this is actually why at Northeastern we had 3 primer pairs on our SARS-CoV-2 tests. The parasites evolve resistance to the tests. So if you, about 10% now in Bangladesh, if you take negative PCR tests and put them under a blood smear microscopy, you'll see the parasite swimming around super happy. If you sequence it, the region of the genome the PCR test is looking for, gone. Right? Because what happens if the PCR test is negative? The person goes out and transmits malaria. Huge evolutionary advantage. Additionally, this is work PhD student in network science Juan He and Tina Eliassi-Rad, who's a professor in Curry, also a member of the Institute for Experiential AI, where we wanted to classify where SARS-CoV-2 genomes have been sampled. Again, we use a neural net for this. Neural net's very good at doing this. And we can get something like 95, 99% accuracy on a multi-class classification, so over like 60 countries. It's beautiful. Couldn't imagine doing a better job. The problem is, why isn't it perfect? Like, it really should be able to be perfect. Does anyone have a guess?
Mutation?
No, it's not a bad guess though. No other guesses? How many people lived in Boston over the summer? 1 person, 2, 3, a few of you. All the rest of you probably came back to Boston. If you tested positive for SARS-CoV-2, did they ask whether you got it in Boston or whether you got it where you're coming from? What do you think the data says? Data says Boston. So what's happening is people in London test positive. They got it in Paris. It's a Paris virus. The neural net knows it's a Paris virus. So the neural net is actually probably 100% correct. The data are just mislabeled. And the way that we actually showed this is Juan came up with this very nice idea to construct a misclassification network. So we get the network of all the misclassified viruses, and we can show that the clusters geographically is associated with the airline travel network, et cetera. So pretty good evidence that the neural net knows what it's doing. The data are just mislabeled. So the reason I'm saying this is a problem for unsupervised learning is if the data labels are just wrong, then it doesn't matter whether you have labels or not. And so for an unsupervised learning task, even if we did the clustering, looked at different clusters, and then revealed the labels, you might think that the clustering isn't working very well because it's got some of these French samples over here in the UK and the other way around, et cetera. Similarly, one of the main things that I do, and a big part of the reason I'm here, is try to improve the way that we mathematize living systems. That's actually how I think we're going to make advances in core AI, is improving the way that we mathematize the living systems, right? Neural nets are supposed to be brains. They don't look anything like brains. They don't learn like brains. They don't use energy like brains. We know a lot more now about how we might actually mathematize what a brain looks like than we did 30 or 40 years ago. What we show in this paper, social scientists are interested in this contagion dynamic called the voter model, which is like, how do I decide to adopt a new behavior? If a certain number of my friends have it, then I'll do it with probability 1, and if they don't, then I won't do it. It can be more complicated than that. We're also interested in multiple pathogens spreading at the same time, like this flu, RSV, COVID that we heard about in the US. We want to know whether they're interacting with each other. It turns out the signature of that interaction is that you'll have additional nonlinearity in the time series. So imagine that there's like a synergistic interaction. This happens with TB and HIV, where if you have a TB infection, you're more likely to get HIV, or the reverse. So you actually see a kind of a lag time and then this explosion in the number of cases as you build up enough of the first kind of pathogen. And that's what happens in this voter model, right? Like, it's very slow to get started, and then once a critical number of people get it, then it just flips over. It turns out that these generate time series that look kind of like each other, these two models. What we showed analytically here is that they're exactly the same model. People have just labeled the parameters differently. And so what we've shown here is that you actually can't tell without a whole bunch of additional information whether something is being generated because of this threshold type process, which we don't think happens in biological contagion, or this interacting contagion process. Lastly, project that we're just getting ready to publish, looking at how, if you were in the US during COVID, you know that a lot of individuals got released from jails and prisons to try to bring down prison volume. If you are familiar with the US prison system, we incarcerate more people than like the next 50 countries combined. It's a huge, obscene percentage of the population, and it's heavily racially biased. However, over the last 20 to 30 years, that bias has been decreasing. It's still obscenely high, but it has been decreasing. Turns out it's quite difficult to get access to data on who's in prison in the US, and technically it's supposed to be all public, but it's like pretty hard to get access to it. And so we had this idea. We said, well, the governors are releasing people. They're proud of this. It's gonna be racially biased. Maybe it's not intentional, but they're gonna release the data because they're proud of it. And it turns out that was the case. And we were able to compile monthly data for 3 years at the state level for everyone who's incarcerated in the US, race, ethnicity, et cetera. And what happened is it was overwhelmingly racially biased who got released. Overwhelmingly white, reversed a multi-decadal trend in decreasing racial bias. Now, why am I telling you this story? One of the reviewer comments, how is race ethnicity determined? Does anybody know? I'm telling you that it was racially biased. How did I decide who was in what race in the data set? You all probably filled out a form when you started at Northeastern, right? When you came through immigration. If you came through immigration,
Self-reported.
a lot of it's self-reported. We called up the state of Texas, and they said, well, what do you mean? We just look at them and write it down. That was their official answer. And it's funny, but it also isn't funny because, of course, as soon as you're entered in the computer as a particular race ethnicity, you will be treated that way by the criminal legal system, right? So the challenge here is that we're trying to understand something about racial bias, but actually each state has a different set of race ethnicities you can pick from. Some of them are self-reported, some of them are not. There's a checklist, but the corrections officer picks it. Again, in the state of Texas, they actually do just look at you and write down whatever they want to write down. So it sounds like we're in a lot of trouble, and I think that we are in a lot of trouble. But I wouldn't be here if I thought we were in that much trouble. In fact, it's exactly why I'm here. So supervised learning doesn't work. Unsupervised learning doesn't work. I know there's a bunch of people rolling their eyes. I cut off part of the slide earlier. Reinforcement learning, which is that basically like I just feed the errors back on the model. What we're doing at Northeastern is sort of reinforcement learning with an important addition. Not only are we thinking about how we adaptively learn through feedback, we also know that when you have humans or labs in the loop, you can dramatically improve the process of going through, evaluating the models, suggesting new experiments, trying to understand bias, right? Because a lot of these things, like whether something is fair or not, can be quantified, but it's also a social feature, right? And so in some ways, you actually have to have the human in the loop. In the case of the malaria diagnosis, how do we resolve that? Well, what matters is whether you need to treat the person for malaria or not. And that's a decision that actually can be made even if the PCR test, the MRDTs, and the clinical workups disagree with each other. Now, maybe not all the clinicians will agree on what we should do, but that is an outcome that we can actually work on, which is how should we be treating these patients for malaria? And so the path forward, I'm convinced, and the Institute for Experiential AI is convinced, and it's part of what's special about Northeastern because they're also convinced, is by tightly coupling human systems, laboratory research systems with AI systems in a way that acknowledges the fact that a lot of what we care about is subjective anyways. And so we need to have humans that are evaluating and interacting with the predictions we're making. Additionally, we can also think about how to advance our core AI capacity by improving the way in which we bring knowledge from systems that AI is supposed to mimic into the way in which we build our algorithms for training, we build our algorithms for evaluating data, we mathematize these living systems. And the only way that we're going to do that is by breaking down the traditional academic silos that exist between the life sciences, the AI faculty, the engineers, the clinicians, all of the people, the government officials, all the people that have to be at the table, the ethicists, the philosophers, if we're really going to come after this problem. And so that's why I'm here. That's why I'm advocating for things like building wet lab in the loop systems. This is an example of work that we've done with Professor Rebecca Carrier, where we take different fish that have different microbiomes, we test them with respect to their response to diet cue. We do what we did with this cancer study, which is to gather a whole bunch of multi-omic data, infer the network structure associated with the microbiome, the neural patterns, the gene expression, the genomics, et cetera, make predictions about how to perturb the system to get a specific phenotypic output. We actually engineer it. We know it's going to be wrong. Why do we know it's going to be wrong? Because these organisms evolved to have phenotypic robustness, homeostasis, et cetera. As soon as you perturb the system, all kinds of other pathways will activate that we previously had not seen before. We will learn about those. We will suggest new experiments and new perturbations, and we will then be able to drive the system towards the phenotypic space that we actually care about. We have an NSF grant now with Dan Bolnick, Tina Eliassi-Rad to do this out in the wild, where they've seeded a whole bunch of lakes in Alaska along a 1,000-kilometer gradient with lab stocks of fish, and we're going to watch as they adapt to this climate gradient and try to understand mechanistically how they're responding, but in a way that acknowledges we don't expect there to be this kind of easy predictability between these different populations. Instead, what you're going to have is something that requires this iterative approach to learning. So, despite the fact that the challenges associated with ground truth in the life sciences are complex and I think nearly unapproachable with respect to many of the off-the-shelf machine learning methods that we have, I think it's entirely approachable by considering, yes, a reinforcement process of learning, but also bringing humans into the loop in a meaningful way, bringing wet labs, field studies, sociological studies into the loop in a meaningful way, but then also further closing that loop by improving how we mathematize systems that are important for artificial intelligence, to include things like the way in which we conceptualize neural networks. There have already been studies showing that if you improve the way the neural networks look with respect to human brains, it brings down the amount of electricity required to train the models. There have been some studies very recently looking at taking these large language models and sparsifying the parameters down by multiple orders of magnitude and still getting the same accuracy, right? So we probably don't need 10 to the 10 parameters. Our brains certainly aren't doing that. But we actually know a lot about how our brains look and work and operate and function. So with that, I want to thank the team at the Institute for Experiential AI who ensured that we had lunch and videos and a room and all of the information that you needed to find out about this. All the people online, really appreciate it. Ricardo, again, for the introduction, and all of you for attending. I think we have a few minutes for questions, and I'm happy to take them now. Thank you.
So if anybody has a question, just raise your hand and I'll bring the microphone over to you. In the meantime, we did get one question from online.
Who's it from? Somebody hard or easy to answer?
This is from Istvan Kiss,
Okay.
who asks, could you elaborate on your findings about predicting epidemics and the intuition behind having more data being not advantageous?
Yeah.
Is it because the model is changing in time?
It is not really because the model is changing in time. That's the easy way to think about it. The way to think about it is it's actually the way we label data is just a problem, right? So we said we had a flu outbreak this year. And we said we had a flu outbreak last year, and a flu outbreak, it's a completely different virus every year. The vaccine worked really well this year, didn't work a couple years ago. We had COVID last year with masking. We didn't really have masking this year. So what's actually happening here is it's sort of the data generating model. It's also sort of the evolutionary process. It's also interacting with human behavior, et cetera. And so the easy way, at least from my brain, to think about it is that the model that generates the data changes in time. That's probably not what's actually happening, but I think that that's not a bad way to think about it. And it wasn't, for example, it wasn't a coincidence that if you paid close attention, the flu was predictable until we got past 1, and then it shot up. Flu happens annually, so it's predictable throughout a seasonal outbreak, and then it's not predictable interseason.
And while folks continue to process, we do have another question from online. This one is asked anonymously. Can you explain the comment you said in passing about, quote, biological infections don't happen via a threshold process?
Yeah, that's a good question. So, well, all questions are good. This one actually just turned out to be particularly interesting. We wrote this paper, and we wanted to come up with some examples of threshold processes. And so the way the threshold would work is like the probability of you getting COVID is 0 unless 5 of your social connections have COVID, and then it's probability 1. Normally, the probability would increase linearly with the number of connections that you have. It turns out nobody has ever actually looked to see whether this threshold effect exists. It's a little bit hard to reason through how it could actually work. It's possible. The bigger point is that it is, if it were there, at least in the time series, it's indistinguishable from lots of other mechanisms. Because basically what you kind of imagine is like, there's like a latent heat that builds up in the network, and then something can sweep through very quickly. And that's what happens with the threshold effect. It takes a long time to get that first cluster of people, and then it kind of explodes. And again, this is why, if you were from last year, one of the Nobel Prizes was given for spin glasses and complex systems. And the reason why is because all of these things I've just been talking about, we could be thinking about as kind of either far from equilibrium or frustrated systems where we could model them with spins. And that's another way to think about what's happening with this threshold effect, also with the interacting contagion effect.
Thank you, Sam. I think we have time for about 1 or 2 more questions, if any folks online or in person have them. In the meantime, I just want to remind everybody about another seminar we have coming up in 2 weeks. That'll be online. The recording for today's talk will also be available on our website. Again, that's ai.northeastern.edu. Thank you all for joining us today. And again, if we have 1 or 2 more
question.
Oh, and lunch is here for the folks in person.
Yeah, so.
Ruben, 1 second.
Oh, yeah. Sure, so in this talk, you mentioned a lot what might be the most productive avenues to move forward. Is a accurate summary of part of your talk also that you should focus on near-term prediction as opposed to far-term prediction?
Yeah, great question. So, was my talk about productive areas, and should we focus on near-term prediction? I should have prefaced this and said it multiple times that this is my biased view on what we should be working on. It's what I came here to do, and I'm also trying to convert some people to come work with me. So there are certainly people who will not agree with this, and you have noticed that I basically only cited myself throughout this entire thing, and so I have neglected a massive body of literature about all of these issues. I took that prerogative as somebody who's giving the seminar and isn't up for promotion or anything else, and no one's going to vote on me afterwards. So if I make everybody upset online, I'll have plenty of time for them to cool off. So yes, I do think these are the productive areas. Yes, I think it's very clear that short-term predictions are feasible. And I actually think that that's probably one of the ways that, and there was actually just a study looking at cancer treatment and how if you sort of make these short-term evolutionary predictions and feed that back on the treatment regimes, you can keep the cancer out of a regime where it becomes resistant. And so I think we can actually play to that advantage, right, where you're really making these fast feedback loops with respect to things like treatment, et cetera. Now, that raises a whole host of ethical issues, which is why we have an amazing ethical and responsible AI team at the Institute. And this is also one of the things that we've discussed quite a bit is like there are ethical challenges associated with mislabeled data, with how mislabeled data are used. I mean, you think about all the issues associated with that prison study that I showed you, and what we need to do as an institute is embrace those. And again, one of the things that I talked about when I gave my job talk was, why would anybody come to Northeastern and do AI? Why wouldn't you just walk across the river and go to MIT or Harvard? Well, the reason why is because if you're going to be any good at this, you've got to have computer scientists, ethicists, life scientists, engineers, all working on the same problem. And so you'd have to go door to door at MIT and pull all these people together, and most of them are going to say no because they're too busy. Instead, Northeastern understood the problem well and brought all of these people in under our executive director, Osama Fayyad, to build this Institute for Experiential AI because they understood that you have to have all these components together if you're going to be successful.
Any final questions?
Yeah.
Oh, great.
Professor, you explained about the research, like in which you have done, like about malaria and predictions. So in that you mentioned about the models, what is it to do that. So the model used there was SVM for the classification, or what? Like feature engineering will be done first.
Yeah.
Then might be some SVM classified might be applied in the later stages.
Yeah.
Predict.
Great question. So the question is, how did I build the predictive model and whether I use this particular method, SVM? No, I did not. We use a random forest model in particular. So a lot of the reason you pick models for one of two reasons. Either they're mathematically convenient. So if anyone ever tries to tell you that they picked a prior distribution for a Bayesian analysis for any other reason besides mathematical convenience, you immediately call BS on them. Additionally, you pick it because it works for what your use case is. So we had to deploy it in JavaScript, and we could figure out how to relatively straightforward do that if we had a decision tree that came out of a random forest model. And so we didn't look at a whole bunch of other ways of predicting. The actual biggest challenge here is this is using symptoms. Malaria presents like a lot of other febrile illnesses if it's not too severe, dengue, Zika, influenza, etc. And so it's actually pretty hard to do this unless you know that there's a malaria outbreak going on. So the real challenge here is actually about integrating the testing data and some of the other types of surveillance data.
So moreover, like we can integrate other models and check like accuracy, because I think like SVM will be more in this classification model. If we use, like earlier I built a model, so I was predicting some cancer, so I used a data set, so in which I use SVM model. So there was about 99% accuracy I got in that prediction.
Yes. So your intuition is probably right.
Yeah.
That in this case, so you would expect this SVM model to work quite well if you think that the challenge is right at this boundary where you have a switch between, say, malaria positive and malaria negative, and what you want in particular, what the objective function is doing is upweighting things that are right at that decision boundary. This is sort of the opposite of what happens for like lasso regression, and that's part of the reason why SVM is so good. So I think you're probably right. I think also I didn't want to have to be responsible for implementing that in JavaScript. So it didn't matter whether you're right or not. It just mattered whether we had the time to implement it, which is not meant to be like a flippant answer. It's meant to be like a lot of the times doesn't matter how good the model is if you can't get it into production. which again, you get said plenty of people online rolling their eyes. You can definitely put SVM model in production in JavaScript. I understand that, but yeah.
Sure.
Rama, did you have a question?
Yeah, I have. Hi Sam. Maybe I'm totally out of AI. I have no idea. But when talking about the time series, so I understand that exactly 100 years before COVID, there is a pandemic called Spanish flu. So exactly 100 years later in 2020, then we all hit the world entirely hit by COVID. So is there anything that AI would have alerted the systems like maybe the FDA or the CDC to know about this before?
Yeah, great question. Also, if once you start giving a lot of talks, one of the things you'll realize is as soon as someone tells you they're not an expert in something, a question that's hard is coming, right? So like that's almost always, you just have to sit there and prepare yourself and like be very serious and know that it's coming. In fact, we think that this is exactly what led to differences in country by country performance associated with response to SARS-CoV-2 is that the US and Western Europe heavily emphasized influenza with respect to pandemic preparedness. And countries in Southeast Asia had experienced MERS, SARS, and they actually had systems that were in place specifically to deal with this kind of thing. In South Korea, for example, that's why the telco data gets released to South Korea CDC for contact tracing because of MERS. And they understand quite well that for a disease like SARS-CoV-2, you have a pretty narrow window of time when you can control it with contact tracing before it's going to get out of hand. For flu is basically so deterministic that as soon as it starts spreading, there's not much you can do except wait for a vaccine. And so what happened is that the US, Western Europe, et cetera, had over-indexed their preparedness on flu and were caught off guard. And what we're trying to do now at the Institute using large language models is learn about what other kinds of plausible pathogen scenarios could occur so that we can better design training exercises for public health officials to try to keep them unaware. The other thing that happened, I was on the phone with some colleagues at Korea Telecom early on, and I asked them, for data on contact tracing, et cetera, and they said, well, we'd be happy to share that with you, but I don't think it will help the U.S. And I said, well, why not? And they said, well, in South Korea, we care about each other, and in the U.S., you don't, and so you're in a lot of trouble. And so I think the other component of all this is like the massive behavioral effect of what happened in the United States during SARS-CoV-2, the fact that we had no culture around mask wearing, but we also don't have much of a culture around making individual sacrifices with respect to societal benefits either. And so it was a huge barrier to overcome. Awesome. Well, thank you all very much. I really appreciate it. Make sure you come to future talks. Get in touch with us if you want to get involved, and have a great rest of the week.
The Institute for Experiential AI is proud to invite you to our spring seminar series, featuring leading AI experts and researchers sharing cutting-edge ideas. You can expect AI leaders from a broad range of industries sharing perspectives on defining and applying ethical AI in our distinguished lecturer series, and EAI faculty and Northeastern University researchers discussing their AI research in our Expeditions in Experiential AI series. Registration is required, and the seminars are free to attend. We hope you'll join us for an invigorating spring semester.